processes that wrap themselves around axons of neurons, okay? Axon of a neuron. What's happening here as the cell wraps itself around the axon of the neuron is that it is basically surrounding the axon of the neuron with several layers of plasma membrane. If you are comfortable with what you've learned so far, the plasma membrane, while it does have proteins in it, is dominated by the phospholipid molecule. And we have made a big deal about how this lipid core brings certain properties to the membrane. The membrane, as it wraps around the axon, eventually loses its proteins. It's really not about transport that we have all this membrane. What it is about creating kind of a fatty barrier around the axon. And that is going to be the subject of some slides that we have coming up. So by wrapping itself around the axon, it is creating a sort of fatty layer around the axon, which ends up being very much like insulation around the axon. If you ever need anything to help you about this insulation around axon, just take a look at electrical cords or whatever, okay? It has an outer covering around it. Or let's say we can go inside, cut it open. We know there's copper wires in there. Each of the copper wires might be considered an axon, but then there's always some kind of an insulating wrapping around it so that we don't get shorts, okay? All right? And it's similar in this regard. So this wrapping is going to be something referred to as the myelin sheath, and it's the oligodendrocyte that does this in portions of the central nervous system. If you're ever searching for relevance, it is the breakdown of this sheath that occurs in CNS of multiple sclerosis patients. The sclerosis is the breakdown of the sheath. The breakdown of the sheath, then, if you know MS patients, at least part of what the MS patient deals with is a lack of control over body parts. Okay? All right? This is the counterpart. We're going to look at these in just a moment, but since we're on the topic of myelin sheath, this is the counterpart of the oligodendrocyte that occurs in the peripheral system. It's called the Schwann cell. This doesn't look like much of anything. There are better pictures of what Schwann cells look like, but instead of a single cell with many processes wrapping itself around multiple axons, a Schwann cell wraps itself around a portion of a single axon. It's also found only in the peripheral nervous system, which probably doesn't mean very much earth-shattering. I guess what it is saying is that we can have myelin sheaths both in the central and peripheral nervous systems. Okay? So that's what the Schwann cell does, and we'll spend more time with that in just a minute, okay? Otherwise, out in the peripheral system, we have another type of neuroglia referred to as satellite cell, and satellite cells are protective cells that surround nerve cell bodies outside of the CNS. If you have more experience with the nervous system, then you know that nerve cell bodies oftentimes reside in the central nervous system, brain, spinal cord. But there can be situations where nerve cell bodies of neurons exist outside of the CNS, in the PNS. When they exist, they are often found with other nerve cell bodies of neurons and surrounded by cells called satellite cells, cells that protect the nerve cell body. Okay? So, satellite cells, similar function to astrocytes, but these are found outside of the CNS, where astrocytes are inside. The Schwann cell, a neurolemmacyte. This should say, well, it says lemmo. Anyway, neurolemma is a fancy way of saying the membrane of a neuron. Okay? We've seen the sarcolemma as a fancy way of saying the membrane of a muscle cell. So a neurolemmacyte is surrounding the membrane, or it has something to do with the membrane of a neuron, if that helps. A long time ago, I didn't learn the term plasma membrane. I learned the term plasmalemma, if that means much to you. So lemma is some kind of, I don't know what the derivation of the word is, but it has something to do with membrane. Now, vital to regeneration of damaged peripheral nerve cells. If you take a look at your book, I don't believe I've posted it in this presentation. If you take a look at your book, you will see that the myelin sheath created in peripheral nerves can actually serve as a pathway for axonal growth if the axons were to get cut. That has some great implications because we know that nervous tissue does not regenerate itself very well, okay? So right up in here, if you were to get into a cadaver and dissect, you're gonna find lots of blood vessels, okay? But then you're gonna find these cords of nerves that go through here, okay? These cords of nerves are usually yellowish in color due to the presence of lipids known as the myelin sheath. And if you were to cut nerves in here, if you were able to put them back together quickly, you might be able to get axonal growth through the tunnels made by the myelin sheath, if that didn't miss you, okay? What it does is it says that there's kind of hope for repair in the nervous system. So take a look in your book and see if you can find that, okay? The neuroglia sure are fun, but the neuron is where it's at, and the neuron is really the focus of what we talk about quite a bit from here on out. How the neuron works is what we're trying to accomplish. How is it controlled? That's what we're trying to do. Don't need to say much about some of these basic things. Structural unit of the nervous system, okay. The axons make up the nerves. Large cells, longevity is interesting, okay? This is one of the, let's say, the great strengths of neurons and the great weaknesses of neurons, is that they live for a long time. On the other hand, if they are destroyed, they are not regenerated. We don't build new neurons. Typically, there are some exceptions to that rule. So if somebody is 100 years old, they have neurons in them that are 100 years old, okay? All right? Amitotic with few exceptions. There are some exceptions to this rule. What does this mean, by the way, amitotic? A means without the ability to undergo mitosis. So no reproduction of these cells. You damage them and you lose them. It's unfortunate, okay? High metabolic rate. This is a very important consideration, okay? I can tell you from personal experience in the last couple of weeks, okay? If these cells are denied oxygen, then unconsciousness results almost immediately, okay? So when somebody's not getting enough blood flow to the brain, boom, they lose the consciousness and they teeter. Okay, so I'm not going to get too personal here, but this is one of the problems my father's had with his dementia, is there are portions of his brain that are not controlling his blood pressure well. So one of his big problems was to get out of bed and boom, okay? Because he's going from laying down to standing up. His body has to compensate, all right? Not enough oxygen to the brain, you lose consciousness. You get dizzy and down you go. This one here does take a little bit of understanding. Right now, most of us in the room are probably running our brains on glucose, okay? It's the preferred energy source for the brain cells, but it's not the only energy source. The problem, if you switch away from glucose, is it takes a couple of days for the brain to do this, and there are maybe some fatigue or some irritability that comes along with it. Basically, the brain prefers glucose as its energy source, not fat, not amino acids, glucose, okay? If you give yourself some time to transition, you can transition away from glucose as the primary energy source. It just takes a little bit of getting used to. If you don't know what I'm talking about, if you go on a low carbohydrate diet, you have to switch away from glucose metabolism in the brain to a different type of metabolism. It can happen, okay? The numbers you're going to see when you look perhaps next semester at the cardiovascular system, and the respiratory system, about 20% of the oxygen in every ejection of blood leaving the heart is used by the brain. About 20%. If that helps to, and think it's only a couple of pounds up there, okay? So this is a very energy involved device. In a room like this, where you are concentrating, you are actually burning a lot of calories at the central nervous system, okay? So concentrate, okay? All of them have a cell body and one or more processes, really true. This is a typical neuron. This is a multipolar neuron. We'll learn that there are some other kinds. Let's take a look at what we've got here. We have a central, a nerve cell body, an NCB, nerve cell body. It has typically a fairly large nucleus. It has endoplasmic reticulum, okay, which is somewhat, this is a minor technical issue. It's somewhat detached from the nucleus, unlike other cells, and as a result, the anatomists give it a slightly different name, okay? Chromatophilic substance or Nissl body. You can just tuck that away if you like, all right? So there are organelles in the nerve cell body, and then there are multiple processes. In a multipolar neuron, there are many dendrites, and there is typically just one axon. However, axons can branch many times. So the cell will have one axon which can be multi-branching and go to different parts of the body, believe it or not, okay? And then the axon itself will eventually divide many times and terminate, okay? Nerve cell body, organelles, dendrites, axon collaterals or collateral axons. Is it on this page? No, it's not, okay? And then axon terminals, yeah.
What makes it multipolar?
Good question. You know, I don't know the derivation because we have multipolar and bipolar and unipolar neurons. I wish I could tell you. Sorry.
It was on homework. It's just a straight guess.
Okay. All right. It was on homework, and there was nothing in the book.
Yeah, it was in one of them.
Okay.
It was 4 hour lab.
Did it tell you why? I mean, tell you why it was called multipolar?
Shoot, now I can't. It was because of the way it was set up. Like it was like one had like one. It was the way it was
Okay.
like built.
Well, we'll see a picture and we'll see a diagram in a minute. Anything to add? Okay. All right. All right. So we see here that this one has Schwann cells on its axon, and we know that axons can be really quite long. Down here we have the things called the axon terminals, and there, as we have learned in neuromuscular junctions in the skeletal muscle system, that there can be multiple terminal points on these axons. A single axon can touch multiple muscle fibers, okay? What do we see up here? The soma is the same thing as the nerve cell body. Soma is a Greek word, I believe, for body, okay? So we sometimes see something, the term soma used, all right? Or we see nerve cell body. Biosynthetic center of the neuron. We find out that neurotransmitters are present at the axon terminals. Some neurotransmitters are actually manufactured up in the nerve cell body and then transferred down there. From what I've read about axons, they have kind of a railway system carrying materials back and forth from the nerve cell body to the axon terminals. So this thing is, these terminals are in communication with the nerve cell body even if they're several feet away. So biosynthetic center means that it's the nerve cell body that really makes the stuff that can exist way out several feet away in the axons, okay? There is the rough ER, which is a protein producer. We actually are going to find out that proteins are fairly important in how this axon functions, and so we have a synthesis center for the production of protein, okay? Most nerve cell bodies are in the CNS, however, there are some exceptions to this rule. And when we get a bunch of nerve cell bodies clustered together, it's called a nucleus. Sorry about all the terminology. In your brain, you have clusters of nerve cell bodies known as nuclei, and you have layers of nerve cell bodies known as cortex. Okay? There are other words that can be used. All right? Right now, it's just a big black hole. You don't know what they're for. You don't know why they might be there. Later, we find out what they're for. Lastly, on this slide, the dendrites convey incoming messages towards the cell body as something called graded potential. I'll lay something out on the screen for you, and then perhaps you'll encounter it as we move forward. The dendrites in a multipolar neuron are typically then synapsed with other neurons. A axon terminal will reach one or more places on the dendrites of this cell, okay? As I told you last time, there can be 10,000 such synapses that surround this cell. Some of those synapses are with the